All right, so welcome to your cell organelle notes, and we're going to take a tour of all the organelles that cells can have via the path of a protein. So when you're looking at the whole sheet, just kind of zoomed out, you're looking at an entire cell. It's not any particular plant or animal cell, it's just kind of a model, stereotypical cell showing you the things that could be on the membrane or inside that membrane, so like within the cytoplasm, etc. So just taking a look on the outside, this line around the outside is the cell membrane. We'll talk a little bit about that kind of up here in the top corner. Just showing you some things that cells can use to move. So there's some cilia here. There's also some villi and microvilli, like the cells that you have in your small intestine to increase surface area. Um, and again, this is not to scale, so this is just to kind of give you a good idea of what's in the cell. So let's go ahead and start with um, the extracellular matrix up here on the top. So I am going to go ahead and zoom in. So outside of the cell is what's called the ECM, the extracellular matrix, and that consists of a variety of different proteins. Those proteins could be those proteins could be collagen or elastin. And we'll talk more about proteins and the types of proteins that you might see later. Or you could have carbs, it could be glycolipids, glycoproteins. Um, those are going to be lipids or proteins with a glycogen group on them or glycogen attached to them, and we'll talk more about those too. But there is a world outside the cell, and that is called the ECM, the extracellular matrix. So if you see proteins outside, that's part of the extracellular matrix because cells are connected not only to other cells, but to proteins in the extracellular matrix, right? So let's go ahead and start to label this. One of those proteins, again, could be collagen. So we're going to go, and I will use this text box just to help speed things up a little bit. All right, so this protein right here is, let's just say this is collagen. Everything here is part of the cell membrane. So the basic building block of that cell membrane is a phospholipid. Remember, you have a hydrophilic head and a hydrophobic tail region, right? So that is a phospholipid. We can label that. These hexagons, or what look like diamonds, they should really be hexagons because they're supposed to be sugars. That's a carbohydrate chain. So I'm going to go ahead and say that is a carbohydrate chain. If that carbohydrate chain is attached to a protein, like down here, then this entire molecule, I'm going to circle the whole thing, is called a glycoprotein. And if that carbohydrate chain is attached to a phospholipid, like it is over here on the left, then that's called a glycolipid. These molecules here in the middle, kind of integrated inside the membrane, is cholesterol, and that's going to help fluidity and kind of prevent the membrane from getting all seized up and locked up. It'll allow materials to pass through, but that's kind of a quick tour of the membrane. So let's drop down here to the nucleus. So this is the nucleus. Again, this is where the DNA is. Really quick, let's just go ahead and go over for the purpose of making a protein and having it taking you a walk through the cell, you are going to have transcription of the DNA into a messenger RNA strand. We'll talk about the details of that later. But again, transcription is going to occur in the nucleus. There's my little text box here. All right, transcription does occur in the nucleus, and actually, and specifically, I'll make a note of this down here. It occurs in the nucleolus, which is the part of the nucleus that builds the mRNA strand. So I'll say transcription occurs in the nucleolus. And that's part of the nucleus. And again, if you are a human, you have 46 of these. Again, these being a chromosome. So if you just look at this chromosome, again, I'm only showing you the one here, but Again, your chromosomes are condensed when they're not being copied. They're wrapped around these histone proteins, which is these circles with the H's in them. So that's why I have histone protein up here, just like a little key so you know what you're looking at. And again, the default, again, if your cell is not replicating its DNA, it's not copying it, it's not trying to transcribe it, the default state of your DNA is to be condensed into what is called chromatin. And again, that is just the DNA wrapped around these histone proteins. That way that you can pack all of that DNA into such a small space by wrapping it around the DNA. Again, DNA is negatively charged because of the phosphate groups, so you should be able to infer the charge of these histone proteins. Maybe you did it, maybe you didn't, but that charge would be positive. So histone proteins are positive, DNA is negative, and that's one of the reasons DNA can wrap around these histone proteins. All right, so once you have transcription, again, we will talk more about transcription later on. This messenger RNA strand is always going to move out into the cytoplasm. So I'm going to go ahead and say, I don't know, sometimes this text works and sometimes it doesn't. I'm going to say always goes to cytoplasm. And out in the cytoplasm, you're going to encounter a ribosome. So that's what this little snowman looking thing right here is. It's the small and the large subunit of a ribosome, right? So the messenger RNA will run right through the middle of this. That is a messenger RNA strand. This is a ribosome. So we'll label those. And again, this is the same messenger RNA strand that was made in the nucleolus. By the way, we're going to talk about translation here, so that is what we're talking about. This is translation from a messenger RNA strand into a protein. And this messenger RNA strand will get read 3 bases at a time. Those 3 bases are called a codon. You may or may not remember that from 9th grade, but that's what they're called. And the mRNA strand is read in sets of 3 bases. So I'm just drawing these sets of 3 to show you that there are many, many sets of 3 codons on any particular mRNA strand, right? So as part of translation, what's going to happen, and again, this will come in more detail later, is that tRNA molecules, which I'm going to go ahead and draw up here, which have amino acids attached to them. That little circle up there is an amino acid. I'm going to abbreviate that AA. And this is a tRNA molecule. Has an anti-codon on it. So that set of 3 bases is called the anti-codon. That will match up with the codon. They are complementary. And as a result, what's going to end up happening is it will drop off its amino acid. This will happen over and over and over and over again. And what you're going to end up building is this chain of amino acids, which you can also call a polypeptide. And just for making this go a little quicker, I'm going to type that. That's what's growing out of that ribosome. And this is the primary structure. Right? The sequence of amino acids is what you're building here in translation. And all of these amino acids are linked by peptide bonds. All right, 2 things can happen at this point. If this emerging polypeptide is needed by the cell, the ribosome is going to make its way over to a chaperonin protein. So I'm going to actually split this thing in half now. I'm going to kind of ignore the vacuole and all this stuff over here. Here's another ribosome, by the way, just so that you can see that. Again, it is made out of RNA, ribosomal RNA, and some protein. So let's come down here. Let's say that if stays in cytoplasm, so if this polypeptide is needed by the cell and it stays in the cytoplasm, it stays with its ribosome. So I'll say stays with ribosome. And it's going to go to a chaperonin. And just for lack of space, I'm going to actually put this over here. This is kind of cluttered at this point. I do apologize. That's what I have for you right now. So here's a chaperonin protein. I'm going to draw it kind of like a little trash bin. This is, think of like a chaperone on a field trip. It's going to help this emerging polypeptide form in a less hostile environment than the cytoplasm. The cytoplasm is filled with enzymes and aggressive pH values that this emerging polypeptide is not going to fold well in. So the chaperonin provides, sorry, there's no E on the end of that, provides an environment for that kind of baby protein to start folding. So this is where the protein can fold and mature into its fully functioning either tertiary or quaternary structure. Right. All right. If this protein needs to be used by a different part of the body and it needs to be shipped out of the cell, it's not going to stay with its ribosome and head to a chaperone. It's actually going to, oops, I don't mean to move that whole thing. It's actually going to come up here to the rough ER. So the second option, and this is the key thing, there's kind of two options. So this is option 2. Let's say that this was option 1. This is option 2. All right. That ribosome, along with its emerging polypeptide chain, will attach to the rough ER. There's kind of like a little signal that tells it to go over there, and we'll talk all about that later. So here's the messenger RNA. And it attaches to the membrane on the rough ER, right? And the word rough is referring to the fact that there are ribosomes attached to this, so it gives it kind of a rough texture, right? Once you're in the rough ER, you can mature as a protein, just like you matured inside the chaperonin. You have a nice kind of environment to fold and make your tertiary and quaternary shape. The same is true for the inside of the rough ER, right? So this is where the secondary and tertiary structure comes from, where the folding happens. This is also, I'm going to go ahead and say that's because this environment is protected. It's a protected environment, just kind of like the inside of a chaperonin. This is also where you might be able to modify that polypeptide. So one thing you can do is you can actually add carbs to it. You can make it a glycoprotein. We'll say add carbs. This is called glycosylation. This is actually also how you get the glycoproteins that we just talked about earlier in the cell membrane. And then this is also kind of the start of this transport journey out of the cell. All right, let me move up here. So these proteins are going to be gathered up into little vesicles. So what I'm showing you now are these little circles are supposed to be proteins. And these little vesicles are going to bud off. It's kind of like a bus carrying proteins. And they are going to move, I'm going to label those, they're going to move along what's called cytoskeleton. So proteins and vesicles, and they're going to move along cytoskeleton. They're not just kind of floating willy-nilly, they are actually moving along cytoskeleton. Cytoskeleton is comprised of microfilaments, which I'll show you here in a second. And they eventually make their way to this outer edge of the Golgi body, right? So again, we're kind of taking a tour. So now we're at the Golgi body. The Golgi body, or the Golgi apparatus, does have two sides to its membranes. There's a cis and a trans side. It's just to help you kind of orient yourselves. Vesicles will enter on the cis side and make their way through all these different membranes. So from cis to trans, and then from cis to trans, then from cis to trans. And you'll work through as many as these membranes as there are in the Golgi. Right, and when you're in the Golgi, think of the Golgi as like a post office. So we are going to, your cells are basically going to attach a tag. It's going to stick a name tag on these proteins and tell them where they're going. So let me say attach a tag with destination. And again, that tag is typically a carbohydrate. So I'm going to say that this is where more glycosylation happens. Sometimes that protein doesn't really get shipped out. It actually will become a lysosome. So that's why there's a direction down here going out to a lysosome. That's a digestive body in animal cells, not in plant cells. And then if it does get shipped, let me get this little hand and drag over a little bit. Here we go. The vesicle coming off of this part of the trans membrane on the Golgi body will follow along some more cytoskeleton. You're going to start to notice a theme. Vesicles tend to move along cytoskeleton rather than just kind of floating around. Again, cytoskeleton is usually made of connective or proteins like actin, myosin, filaments that we'll talk about throughout the year. So here are our little transport vesicles with mature, fully tagged, and ready to go proteins inside. And then they can get released because these vesicles will fuse to the cell membrane and release their contents. Right. So that's kind of a tour of that part of the cell. Let's look at some of the other parts towards the bottom. Let's go down over here. So let's talk more about those microfilaments in detail. This kind of set of tubes, there's one tube and another tube here. This is the centriole. Centriole is used for animal cell division. It's going to produce spindle fibers. We'll talk more about that later. A lot of microtubules. My pencil. Your microtubules are going to be made out of a protein called tubulin. Right? Not a very creative name, but it tells you exactly what it is made out of. For fun, this is made out of, it's kind of like a fun fact, this is made out of 2 proteins. So it does have quaternary structure. 2 proteins, I'll say quaternary structure. These essentially act as tracks for the vesicles to move along. And then I'm also going to, well, I'll type this one, it's a little bit longer. Oops. I'm going to put it up here, but I am talking about all of this, the microtubules. I'll say move chromosomes in cell division. Say spindle fibers. So really, I know it's kind of in the smoothie R here, but it's really talking about the microtubules in the centriole. I believe over here, I'll slide over here, another kind of chain of proteins that you might see in a cell is called microfilaments. These are made of actin. Actin is another protein. So all of these little circles is an actin protein. Go ahead and label that. Actin proteins are 2 rows intertwined. And I think just as a key, anytime in this picture you see this little circle, there we go, this circle, that's referring to tubulin, and actin are these little fibers here. So even though that's a circle, that's really talking about actin. And again, here's some actin filaments, just kind of connecting organelles and giving the cell structure. So down here, I am going to label this. Let's label this tubulin as a reminder. Tubulin is what's going to make, come on. Tubulin is going to make microtubules. And actin is going to make microfilaments. And also, that's going to be responsible for helping hold the cell shape. All right, while we're down here, let's take a look at the smooth ER. The smooth ER is for lipid synthesis. It's smooth because ribosomes don't attach to it. There's no reason for them to. They don't get modified and they don't get folded and glycosylated in here. So this is all for lipid synthesis and detoxification. So those are right over here. There's one way, there's the second way. Some lipids that you might be familiar with, bracket those. Testicles and ovaries are going to make sex hormones. So a lot of your hormones, which are lipids, are going to come from cells high in smooth ER. There's going to be a lot of smooth ER in those cells because they specialize in making those hormones, testosterone, estrogen, etc. So I'm going to say hormones. Liver is going to help synthesize cholesterol. Remember, cholesterol is a steroid, which is a lipid. Detoxification of drugs and poison, etc., is only in the liver, only in liver cells. Liver cells. If we were in person, I'd talk a little bit more about Tylenol and aspirin and stuff like that, and I'd write it on here. But basically, if you have an over-the-counter drug or prescription drug, and it tells you that the maximum you can take in any amount of time is so-and-so pills, it's basically telling you how quickly the smooth ER in your liver cells can break down that particular drug. So fun fact there. I am going to go ahead and continue. Let's see. As we move our way to the mitochondria, here's the lysosome that we kind of talked about earlier really quickly. It does have a lower pH because it is digesting material in animal cells. It is performing hydrolysis, right? If you're digesting something, you're hydrolyzing it into monomers. So you guys should have been able to pick up on why it's hydrolysis and not dehydration synthesis, since you are breaking things. Right, and therefore you have specialized enzymes that are proteins that were made in the rough ER and shipped out into the cytoplasm with their little vesicle to become the lysosome, right? So those are hydrolytic enzymes that do hydrolysis. Peroxisomes are very similar to lysosomes in the fact that they are also, they're digesting material in the cell. While we're talking about kind of in the cell in general, remember your cell cytoplasm does have pH, which means you do have a concentration of hydrogen ions, and that concentration, again, is 10 to the negative 7 molar, which is 0.00001, right? Which means the concentration of hydrogen ions in the lysosome is 100 times more, right? So times 10 to the negative 5th molar. All right. Let's go over here to the mitochondria. Here's the mitochondria as a whole. It is a double membrane structure. We'll talk about endosymbiosis a little bit later into why that structure is the way it is. But you know that a product of cell respiration is CO2, carbon dioxide gas, and also ATP, just cell energy. It's actually a modified nucleotide. We'll talk about that later too. But the mitochondria prefers to use glucose as its primary source. And we'll talk about this more later, but it actually is going to use something called pyruvate, not glucose directly. So its preferred energy source is something called pyruvate. I'm going to block this off and put a number 1 by it because it's preferred. But glucose is responsible for getting you pyruvate. Glucose gets into the cell because there are these glut proteins. We'll talk more about glut later, that allow glucose to move in from the bloodstream. So this is the kind of the primary pathway for your mitochondria to make ATP. Glucose comes in through glut. Again, details to come. It gets transformed into pyruvate. Again, we'll talk about that later. And then pyruvate can be used to make ATP. If you are running low on glucose and pyruvate, the second option, so I'll say number 2, is for your mitochondria to use fats. If you are running low on fats, the third option is for your mitochondria, so I'll put a 3, to use amino acids. I'll just say AAs for short. It is possible for amino acids to be brought in to cells using a protein channel, so I'm going to go ahead and label this as a protein channel. We'll talk more about protein channels and pumps and all that stuff in our next video, but just kind of showing you the outline for right now. So here's a protein channel, and amino acids can come into the cell through that channel from the bloodstream. Right? There's also oxygen out here, there's also glucose out here. I'm just kind of focusing on the things that the mitochondria will need at the moment. Right? All right, and I believe, let's see, we can add, that's that. Let's go find a spot over here. Let's go kind of back over here down by the nucleus. Let's just add the peroxisome, because I did talk about it, but I never added it. Peroxisome in cells is going to break down lipid, and that is a little organelle here, so let's go ahead and put a membrane around that. But that is kind of a brief overview of this side of the cell, and we've kind of taken a quick little tour. On the back, I'm not going to spend too much time on it. I think you can, on your own, kind of walk through this and differentiate some of the different structures, for example, in a eukaryotic plant cell or a prokaryotic cell here, like a bacterial cell. The only thing I want to address really quick is that if you zoom in on a bacterial cell, what you're going to see is this very, very high degree of infolding of membranes. Right? The bacterial membrane is not this kind of nice linear circle. It's actually very, very highly folded, which is what we're kind of trying to show you down here in this microscope picture. And the whole point is to increase surface area and locations for reactions to occur. Right? It's actually the beginnings of kind of this multi-membrane kind of pre-eukaryotic organelle structure, evolutionarily speaking, and we will talk about that a little bit more, but I did want to point that out. Just it's not something that you would necessarily pick up on without me kind of showing you, because there's nothing else on here kind of pointing it out. But everything else you guys can look over, just some basic characteristics of prokaryotes. You should be able to identify all of the organelles that are present in both of these cells. Not too many going on in the prokaryote, but certainly you should be able to identify all of these cells. Again, that is a central vacuole in a plant cell. That's not the nucleus in a plant cell. And I hope this helped, and I will see you guys next time.